Thank you very much. It's Happy New Year to everybody, and it is nice to be back here to the lab. And what I want to do today is to demonstrate where we are today with the cryo-electron microscopy. In the area of cryo-electron microscopy, it turned out this is I call a multi-scale imaging on bioimaging. And that span actually a number of technologies from X-ray crystallography, cryo-electron microscopy, optical microscopy, X-ray microscopy, and other types of microscopy. And this actually span across a very broad range of resolution scale. And what we have been doing in the last many years is to push the technology using an electron microscope as an instrument to collect images at a resolution between 3 angstrom all the way to 100 angstrom length scale. So what I'm going to talk to you today is to illustrate with some example how this electron microscopy can be useful at this length scale. Now, in terms of the methodology, this is a workflow diagram. What we do in cryo-electron tomography, we need to do some form of biochemistry or cell biology to get the specimens. Some can be very sophisticated. Now get to the point, I begin to realize we have to do sophisticated method as the X-ray crystallography. So I'm talking to Andre this afternoon that I personally have a long way to learn, but that is where the field goes. We really need to spend some time in the front end. So provided you have the material on hand, the next area is to prepare the specimen. The word cryo-EM means you prepare the specimen in the kind of low temperature and in such a way that you keep the specimen in frozen hydrate that stay at the buffer condition that the biological experiment you are interested in. And then after that, you put in the electron microscope. And what the electron microscope provides you is a two-dimensional images of your three-dimensional objects. So if you have a single particles, that means you have an individual image, a 2D image of the single particles. And so you have a stack of images from different specimen area. What the computer processing is to combine each of these single particles to generate a three-dimensional density map. I mean, from a physicist's point of view, they call it Coulomb potential function. That's exactly what we get in 3D. And then after that, if the resolution is good enough, we can build model of individual molecular components, and at the end of it, you deposit in the electron microscope database and the protein data bank. So as in this pipeline, a lot of these steps are highly assisted by data processing. So the computer control of the instrument and also data processing to convert from 2D image and 3D images and eventually build model are very, very computationally intensive, more so than the traditional X-ray crystallography. Now, you may ask, where are we in the field? So here is a statistics about the structure has been deposited in the protein data bank and also in the electron microscope data bank. And the one in green are the numbers of structures that has been deposited in the density map only. Now, some of the density map actually have associated model are represented in the bluish color. I get the other way around. The density map are represented by the bluish color bar. So right now we have more than 3,000 structures have been deposited in the electron microscope database, and less than 1/3 of them actually have associated model, or you call coordinates, that deposit in the PDB. And you can see in the last 5 years that has exponential growth in terms of the production of cryo-EM structures. Now, the next question you may ask is, what resolution are we resolving in all these structures? The next slide shows you a dramatic growth in the last few years. In 2008, we were only one of the very 2 or 3 groups that able to get to below 4 angstrom resolutions, which the protein backbone can be traced. But in the last 2 years, you can see that's a big jump in terms of number of structures solved not only in our lab, but many labs around the world as well. And dramatically speaking, just looking at the map of the structures at 3.5 to 4 angstroms, there are about 4 times more structure done in 2015 than 2014. You can imagine this trajectory. I think it's going to grow. It's not going to plateau. I expect that it's going to grow. And so then the question is, what kinds of specimens have we been looking at that can give you these kinds of 2 to 4 angstrom resolution? In our center, which has been supported by NIH, we have solved more than a dozen structures that we're able to trace their backbone. Many of them are virus structures, the other are molecular machines, and I think a couple months ago we published a structure on a membrane channel. And so there are really a variety of molecular machines that can be done this way. And the question is, why all of a sudden that things seems to be much easier? Now, there's multiple reasons for that. One, I would attribute to the development of the direct electron detector. In the old days, we collect images in a Kodak photographic film, and subsequently we collected on a CCD camera. In the last 2 or 3 years, then we can collect the data in these direct electron detectors. Direct electron detector is basically a very expensive CMOS detector. The physics is very similar to the iPhone, but it costs about $600,000 or $700,000 a piece. So that's the only difference. Of course, there are more sophisticated electronics that behind each of the pixel that allow you to have really, really high detected quantum efficiency at all frequency when you record the images. And so the second attribute to this success is the available software from many labs. We develop a lot of software. We just let people use it. I just discovered people use our software to compete to myself, but that's the way how it works. And so I think that really provides the community an open source code that they can actually readily when they have the data. And the third one that I think become more important in the last 2 years, that we have developed more rigorous computational protocol to validate our structures. I think later in the next few minutes, I'm going to show you how we're going to do that. And then, now it turned out that where's the specimen? It turned out a lot of these high-resolution structures actually were done in collaboration with the protein crystallographer, because protein crystallographer have a lot of very, very good specimens that cannot be crystallized. Or with different forms, it is much challenging to do crystallization. So I think these technologies turn out to be a paradise for crystallographer, not because they fail to do things, but they just use an additional tool to enhance the information content. So if you look at all the Science and Nature papers, many of them are actually driven by the crystallographer because they make good specimens. And then they seem to be those are really ready in the refrigerator. Just take it out and snap the picture, and that's here go a Nature paper, right? As simple as that. And so now, given that, then my title say Cryo-Electron Microscopy Molecular Machines. I don't know what the background of the audience. I just want to introduce the definition of molecular machine. The term molecular machine was introduced by Bruce Albert some years ago. He said, we have always underestimated cells. The entire cell can be viewed as a factory that contains an elaborate network of interlocking assembly lines, each of which is composed of sets of large protein machines. Why do we call the large protein assembly that underlies cell function protein machines? He answered, precisely because, like machines invented by humans to deal efficiently with macroscopic world, this protein assembly contains highly coordinated moving parts. That means it's more than one type of protein. There will be multiple proteins and multiple molecules. They have to be coordinated well enough, just like the man-made machine, to make it function. That's why Bruce Albert introduced the term molecular machines. And so now there are many types of molecular machines there. I'm going to give you one example. It turned out to be a virus sample, which you may not think that investigators like me in the medical will work on it, but that's a virus, has nothing to do with any disease at all. And so it's called double-strand DNA bacteriophage. It's a virus infect bacteria. Now, why is it important? But I think in this setting, probably most of you understand the importance about the biosphere. And it turned out these types of virus has been estimated more than 10 to the 31 particle exist now in the biosphere. So it's the most abundant form of life. Just pure number. I think is interesting enough to understand it, right? And secondly, in the biological function, it plays a critical role in maintaining the population ecology of environmental microorganisms. It turned out this virus contains a gene not only code for the protein that make the virus, but it also contains a gene of its host. So in order to get some of the hosts to maintain its ecology and population, the virus is actually a very good media to accomplish that. The third reason is, needless to say, this is a tremendously good model system to understand protein folding and virus assembly. Now, recently, there increasing interest in bacteriophage because bacteriophage kill bacteria. How do we kill bacteria as a doctor? We give it to some drugs, but a lot of drugs is immune by the bacteria. So as a result, one of the alternative therapy is just using the phage as a drug to kill the bacteria. So I think that idea was first initiated by some Russian scientists, but now actually being picked up by some of the physician scientists under the NIH support as well. So I think after all, it may not be that academic, that maybe actually have medical relevance. So that is the justification system I'm going to introduce in the next few minutes. Now, so this system has been studied thoroughly by genetics and biochemistry, which means it's well known how these viruses are being assembled. First, the virus is the gene. The gene is synthesized, replicate inside the host. The host gene will make the protein. The protein will assemble into a phage particle. So what is being assembled is really equivalent to the buckyball, as the nanoscientists would call it. It has 60 copies of repeat, and in this particular phage, it has, I represent by 4 different colors. There is a capsule shell that are arranged like a buckyball with icosahedral symmetry, and it also have this purple color thing called scaffolding protein. And in one of the 12 vertices, there is a particular protein which allows the DNA to encapsulate. In addition to these yellow proteins, there are black protein and many other proteins that actually involve the packaging of the DNA. So when the DNA is encapsulated, and the additional tail fiber is attached and become infectious. So the molecular biology has somehow worked out this pathway of the life cycle of this bacteriophage through very tedious genetics. So later on, I will show you, perhaps using cryo-EM, just a few such experiments, we can actually map out the entire pathway without doing genetics as well. So that will be the later part of the talk. So what I'm going to show you is the structure initially, how using cryo-EM to study a structures of the mature phages. And so here is the image. This is the first frame of a direct electron detector image. Now, one thing I haven't mentioned to you about this expensive detector, it has a very high frame rate. That means you can take multiple frames in a very short time of the same specimen area. Unlike a CCD camera photographic film, you zap some electron and you get one image. But here you can get multiple frames of images as a function of time. So the frame rate is 60 frames per second. Now, one of the challenges in doing biological specimen in electron microscope are twofold. One is the specimen get damaged by the electron beam. Second is the specimen actually move under the beam very quickly. Okay, the specimen movement would blur the image. As you can imagine, you want to take a picture of me. If I keep moving, you're not going to get a high resolution image of me. So it has to be stationary. So it turned out using this detector, because the ability that you can do multiple frames, then in each of the frames, we can align it one frame with respect to other, so you can make a potentially blurry image into a sharp image. So I think that is, if we call the revolution, is one of the two important reasons to do this using this expensive detector, is you can get initially very blurry image in the past. Now we can get very sharp image because you can pre-align the images before you do any data processing for reconstruction. So now, as you can see here, is really nothing at all, because that will be equivalent, kinds of first frame of the image. Now, I'm going to show a movie which is an increasing number of frames after I realigned it. As you can see, as I have 5 frames, 6 frames, something will pop out because the statistics start building up, things get sharper and sharper and sharper. So as you get increasing numbers of frames, and things become more obvious, okay? So, and with that frame alignment, and also in our algorithm, we actually compensate for the radiation damage as we take more frames from the same specimen. So we're taking care of that. And so these are the kinds of data that provide tremendous preservation of information content. We can measure the information content actually by doing a Fourier transform. We can evaluate both either real space or the Fourier space. And so these are the images we are getting in these types of electron detectors. Now, with that, how do I know this is actually improving? So one way we do is we do a power spectrums of these kinds of image before we do the motion alignment and after the motion alignment. So here is the power spectrums of such an image. On the left, on half of that, these are experimental data, these are simulated data, and you can see that before any motion corrections, the data can be kind of barely reach the 5 angstrom. But after the motion correction, the panel on the right, you can see data go all the way to 3 angstrom resolution. I think that means our data are good, it's always there. If we don't do the proper data processing, we just lose it. So I think these detectors allow us not to lose it. The data go all the way to 3 angstrom resolution. So that is the kind of quality that we are getting in these kinds of detectors. Now, once you have that, we have many of these particle images, thousands, tens of thousands. Then we start the data processing from a 2D image to a 3D density. And so the way we normally do is to split our data into 2 halves. In other words, if we have 20,000 particles, we get 10,000 particles in one data set, another 10,000 particles in the other data set. Initially, we don't rely on any model because we don't have an X-ray structure of our virus of interest. So our program will build an initial model, very low resolution model, icosahedron and low resolution, just like a football. Okay, not American football, the real football, round. But then you do these alignments and refining the particle with more and more particle, feature will become more and more. So now we have eventually, at the end of the analysis, we have 2 density map. Now I will get later on why 2 density map is important, because from each of the density map we can build model and we can actually estimate how similarity of these 2 maps are to get us to make an estimate of the resolutions. So here is our definition of resolution. In the absence of crystal, we don't have spots. So how do we define the resolutions? The way we define it is using these particular term, these kinds of parameters called Fourier self-correlation, is the Fourier transfer of one map, the dot product with the complex conjugate of the other map, and normalize it. And then as you can imagine, you can see in here, at low frequency, they match very well, up to 1. One map identical to the next map at low resolution level. Now at high resolution, where the noise is, where the thing becomes less resolved, and that becomes the decay. So it has been proven that this will be the right parameter, namely the phase become random at this kind of resolution. So based on this criterion, the map that we are able to generate from the image I show is around 3.3 angstrom resolution. Now, based on this criterion, you ask, well, you get this curve, looks kind of okay, but how do you know you do it right? In other words, you do this data processing very complicated. How do I know you're not over-refine your map? So we have developed a procedure to make sure we don't over-refine our map. One is to randomize the phase of the raw particle images at a low resolution. Say at 3.3 angstrom, we would say randomize all the phases of raw images at 5 angstroms. Okay, that means in my new data set, there should not be any structure that I can see beyond 5 angstrom. Now, if I can get a curve beyond 5 angstrom, I would say I over-refined it. I'm doing something wrong. So this is a curve I'm showing you in the red curve. Is after I randomize the phase beyond the 5 angstrom, the Forest-cell correlation fall off exactly at around 4.5 angstroms. Okay, that suggests to me the map I present to you was not over-refined. So that's one criterion now being adopted in the last 2 years in the field, that to make sure the map we generate are not over-refined. Okay, because image processing can be a very dangerous activity. You can just refine the noise. You can actually get something out. Okay, that's the danger. So that is why I think these types of procedure to eliminate any possibility that we are over-refined and interpreting feature which just come out from noise. Okay, now, then the next question, at 3.3 angstrom, what would you expect to see? So here is the whole map of this virus particle, and I'm going to show you this movie, which you can see there is a hexagon-looking feature, there's a penton-looking feature. It's one asymmetric unit of this virus as 7 polypeptide. Okay, that means 60 copy of the 7 polypeptide. The things are well resolved enough, we can actually delineate this yellow protein, overlap on top of the red protein, and then underneath the green protein the next to it. So we can actually delineate the molecular boundary, each of the protein subunit in this asymmetric unit. There's 7 asymmetric unit per, there's 7 molecule per asymmetric unit, and there's 60 of them. So 60 times 7 is 420 protein molecules that make up this virus particle. So this is our density. Now, from this density, the biologist is just like crystallographer, interested to see where is one end of the molecule, they call N-termini, to the other end, they call C-termini of the molecule. So that is our next step called model building. So here I'm showing you one of the 7 polypeptides that we have. I just want to make sure to mention this 7 molecule is chemically identical, but they are not necessarily, in fact, they are not structurally identical. Okay, because that is, again, a common theme of molecular machine. They can make up the same protein in a complex, but they don't, even though they make up the same chemical species, but their structure can be slightly different. So we're building model for each of these 7 molecules in asymmetric unit. So here is one, obviously, is one end, the N terminal. So I'm going to walk you through from one end of the molecule to the other. So this is the N-termini. So we build the entire polypeptide backbone. In fact, we have developed a software called PathWalker based on this skeleton space, mathematical space, allow us to trace the connectivity, the density. So the map is good enough that actually can trace the density from one end of the molecule, from the left to right, and from the top to the bottom, and then now back to the left, and then get to the right. And so you can see that we can actually build the entire polypeptide, 425 amino acids polypeptide, build the backbone. So now, once we build the backbone, the next thing we want to do is where are the side chains? As you can see, there are bumpy densities sticking out through the backbone. So how do we build the side chain? So the next step, what we did was to use the de novo backbone model and then plug it into the crystallographic software called CUT, and also the CUT and Paul Adams Phoenix, right? Software, so that we could refine the side chain. So here is what we do in this first rigid body and start building a feeding both locally and globally, start building individual side chain and fit the side chain model to the density. And so that's how we finish the entire model of each individual protein. So this gives you a sense of each one of these 7 polypeptide in an asymmetric unit. The model actually fit quite well with the density. Keep in mind the density are I call measure density derived from the images. And now, but then after we get that, we raise another question. How do we know this density in the side chain are actually real? Right? So we need to prove that it's not something that from the noise. So we did the next experiment to validate the side chain density are actually real. So the way we did it was as follows. So we first extract one of the subunit, which we have the density, we have the model with the side chain. The next step we did was to strip off the side chain completely in the model. So it's just like that. Okay, and use that to feed that back into the map, and then compute a map, and use that as an initial model to refine the parameter of each of the particle, 20,000 particle. And from there, then we go back to the raw data. Initially, the first cycle, we don't see the side chain, but after many, many cycles, we get back the side chain. So in this particular experiment is to show that if I give you the most favorable circumstance, I know the backbone, whether using our refinement procedure get the side chain back. And the answer is yes, the side chain does look like what we had initially. So I think that helped us to confirm ourselves what we observed the side chain are not noise. Okay, and so at the end, we have the final image map. And so from there, I would like to show you some of the really good looking side chain, which are the bulky side chain. Here is the backbone, and I'm putting the density with all the aromatic side chains in here. And you can see how good this match. So basically, all the aromatic side chain model fit very well with the mesh density throughout the entire polypeptide. So I think that was the first thing we look for, how good they are. At 3.3 angstrom, we actually visualize the rotamer of the side chain quite nicely, as demonstrated in this particular movie. And so having done this, then we ask the next questions. Do all the side chain look as good? Because there are 20 amino acids. So then we're looking at then the density value at every side chain. And interesting enough, this movie I'm going to show you, the density value not all the density value of all the side chain are equally good. So this movie to show you some side chain actually are not as good. And okay, so here, some of the, of course, in this case, the N-termini seem to be missing in our density map. One residue is missing. Some other good. And I put the color scale. The pink color represents a really low density value. The blue color are good density value. And I'm going to move into a regions. There's some side chain right here, okay? Pinkish. And these also pinkish right here. And so there are side chain density are just not as good. And when we examine what these side chains are, it turns out these are all side chains carry negative charge, like aspartic and glutamic acids side chain. Two of these side chain density are particularly with low density value. That triggered us to be curious why that is the case in this negative side chain has such a low density value. So then in here, So here are the representative of 20 amino acids, these both aspartic and glutamate. There's no side chain density, even though we have the model, okay? And then the rest, they actually match quite well. No, so there are two possibilities we think that may cause that. One, maybe these negative amino acids are very radiation sensitive. If you look at the literature in protein crystallography, some of these negative side chains are very, very radiation sensitive. So then we look into the possibility whether that is the case due to the damage. So here, then we plot all the side chain density value for 20 amino acids, and we discover that since we can take a lot of frames, so we use only the first 6 frames to get the map, and then build the model and look at the side chain density. And we find, yes, that's true. The aspartic and glutamic acid have the lower density value. Now, since we can take multiple frames from the same specimen area, we can also generate a map from frame number 7 to frame number 12, which those specimen has been subject to more radiation damage. So the next slide shows about the ratios, the changes of those density value from the first map, from the first 6 frames, and the map of the model from the subsequent 6 frames. And you can see the changes are not that much difference among all the amino acids. So I think this experiment, radiation damage study, suggests radiation damage cannot explain fully why the negative charge density are low. So the possibility 1 is less likely. Now the possibility 2 is when you think about electron, they're negative charge. The negative charge residue as negative charge. Negative and negative, what do? They repel, right? So if you think about the freshman physics of the scattering, you may like to conclude that because the scattering property and negative, they repel, so they would not be constructive interference, so density would be down. So it should be a negative density. And in fact, you look at the table, International Crystallography table on electron scattering, and you can see that actually it is different between a neutral atom, positive charge atom, and negative charge atom. Okay, so that suggests that it's likely that due to this charge property of the atoms would give you a different density value. Now, when you think about that, it's actually very exciting because in some of these amino acids, they may have different charge states. Okay, so that means from our density map, we may be able to derive the charge state of the molecular species in the molecules. Okay, so I think that potentially be a special niche in terms of electron scattering versus an X-ray. Now, from here, since like the crystallographer, I often get the question by crystallographer, can we refine your map? You know, because with crystallography, what they do is they have the spots and they get the phase and they build the model and then they refine the phase again and so on and so forth. So far, I trust my image, go all the way to the map, to the model. I have not gone back to refine my initial map. The problem is I don't want to be model biased. You know, I build the model, if I do the model and I refine it, I bias my model. Now, crystallographer was very clever. They developed these R-free, kinds of mechanisms to make sure they are statistically relevant. So in our case, we want to build a model to generate a map in such a way that new map can be used to refine the raw images again. But that map better be a correct map. Now, keep in mind, if we build a model like what I showed you, but when we use the model, calculate the map, we will calculate the map that fit the model perfect because you start with something perfect, the map is perfect. However, our experimental map, all the negative side chains are missing. Okay, so that means whatever in the PDB now, whatever people now use in the literature, calculate from the model to a map, that map does not agree with our experimental map at all. However, since now we are aware of this problem, we actually, using the Phoenix, we actually calculate the B factor and occupancy in such a way that we can account for the amino acid for the negative side chain has a different B factor in such a way when we calculate the map from the weighted model, it matches very well with the experimental map. So from that particular types of approach, I think now we can begin to a stage similar to the crystallographer. We have a model, we have weighted accordingly in such a way we calculate map with which we can actually go to refine our raw image again. Hopefully will improve. And in fact, when we did it, our map might get much cleaner. Okay, we don't improve the resolution in that case, but we actually clean up the map quite a bit. The noise in the map and so on will be minimized. So now, when we finish the structure, we did like what the crystallographer does all the time, is to go on to do these some statistic how good your models are. So these called mole property plot. And so it turned out in terms of the crashes of the atom, because no two atoms can occupy the same space, and we are ranked about 97 percentile. This is kind of software available in the PDB. So when we do it, we rank ourselves in this particular structure we did in the 97 percentile. That means it's a solid A in terms of score. Now there are many different scores. One of them, they usually are important, is a mole property score. We ran 100 percentile. That means in the structure we saw a 3.3 angstrom resolution. We compared to all 3.3 angstrom resolution structures saw by X-ray crystallography, we are among the best. Okay, so I think this is where the state of art is. In fact, when you use this criteria, many of the cryo-EM map actually rank quite highly. One thing you may not be surprised because we actually get the phase information, the map actually even not as so-called high resolution as it could be, or it might be, but it is very accurate determinations. I think what I show you here are information which is we developed to validate the map and the model in the most rigorous way. So in other words, from now on, we can actually deposit a model which can be reproduced, the experimental observation, can do refinement, can do the same kinds of high standard as what the protein crystallographer does. So I think that concludes the first part of my talk, where the field is at this level of 3.3 angstrom. I would say in the last year, there are numbers of structures exceeding this resolution level. Water molecule has been seen, and side chain is better looking than the one that I showed you as well in other systems. Now, once we do this and we ask what kind of biology we learn from these kind of structures. So the first thing we learn is when we look at the sequence level, this virus protein does not look like any other protein in the gene bank. That means at the sequence level, we cannot find any similarity. But when we look at the protein data bank, we find two proteins that actually have similar fold. One is called HK97 that has been solved by X-ray crystallography. The other epsilon phage, which also solved by us in EM. They actually have a similar fold with a long helix, a loop, and beta strand. And the only difference is they have different amino acid numbers. And so then the question is how they differ. So now if we put that in the biological context, in the case of the Hong Kong 97, in order to keep this protein intact in a phage particle, the protein actually have to do chemical cross-linking. They have to do chemistry itself to stabilize the capsid. In the case of epsilon, the additional protein, we call stabilizing protein, to stabilize the capsid protein. In our case, there's no cross-linking, no stabilizing protein. So that means there probably there are different chemical interaction to allow one virus to be one size of genome and without any accessory protein. So that means the lesson we learned is if we want to generate a new DNA-containing machine, what would you do? You use this protein fold. Because the next knife after structural biology, I would make consider to be synthetic biology. You want to synthesize different types of molecules or machines to do the same thing better or worse or two different things. So I think what we learned from here is they use similar fold, but they use probably slightly mechanisms to keep this machine together. Now, in order to understand further, we start looking at the molecular interaction between the subunits. So I'm going to show you two movies to see how this interaction occurring. So here is 3 asymmetric unit. I'm looking at the 3-fold here, and I am turning this density map to the model with looking from inside. Now, what I'm showing here are the N termini of the subunit between adjacent proteins. It turned out these N termini seem to form a beta strand interaction. So that turned out to be very unique in our system compared to the other phages, that these N termini of the adjacent subunit across the capsomere, across the hexons, to form that interaction. So that may be one important force to keep this particle intact without additional protein of different types or without chemical cross-linking. And so now, and then the other thing that we discover is looking at the protein within a capsomere. By capsomere, like a hexon, we're looking at the interface between the subunit. So here I'm showing you again in the model, and we find this very intensive interface between the adjacent protein, and in addition to the hydrophobic interaction, we actually discover the egg-center interaction that involve a salt bridges. So for example, like this one, glutamic arginine, again I'm showing the model with the density to show these types of interaction. Now, so this is a 3.3 angstrom resolution. If we measure the distance and the gravity of the side chain, this seems to be the right distance for the formation of the salt bridges. So we discover there's X salt bridges between the adjacent molecule that form this capsomere unit. So that is the kind of structural insight we can get on. Now, there are more detailed things that I don't want to bore you about, but there you can learn, if one is interested, one can learn more about the monomer interaction that form these kinds of capsid particles. Now, so since I pointed out at the beginning, we could look at virus particle. What I show you is this mature virion. But what happened before the DNA is encapsulated? So we can repeat the same experiment, but before the DNA is encapsulated. So we actually solve another structure with the procapsid. It turned out this procapsid is about 100 angstrom smaller in diameter than the mature virion. And the biological question is how they accomplish this expansion when the DNA comes in. So then when we compare the structures, I'm showing you is only one asymmetric unit. I'm morphing this asymmetric unit between the procapsid. Now, in this case, the procapsid, there seems to be a big opening. If I start morphing these from one state to the other, now I just want to make sure to mention that this is not the exact trajectory of the changes. I'm just saying morph from one state to another, and there's quite a bit of structural changes. This N termini is changing, and this hole is kind of closing up. The molecular interactions is different. And now if I'm looking at the side view of this, and so that actually because of this conformational change, the protein thickness in making the shell actually get thinner in the mature virion versus the procapsid before the DNA is encapsulated. So what we learn from here is there is a conformational change of the protein during the maturation process when the DNA is packaged. That conformational change does not involve proteolysis. It's not involve only one domain. It's actually involve multiple segments of the polypeptide that accomplish in such a way that the central hole of the hexon is closing up. The interface between subunit and get more intimate during the maturation process. So I think this all can be done at a kind of 3 angstrom level without crystallography. Now, the other question you may ask is, I show you this very high resolution captured cell. I also pointed out at the beginning, we have this very important protein apparatus, the DNA packaging. Where is it? Well, I don't show you there because during my whole data processing, I assume only icosahedral symmetry for molecules that don't assume icosahedral symmetry will be washed out entirely. So in order to do that, we actually can, looking at inside, we start looking at the additional density in the interior surface, the scaffolding protein. And then we discover the scaffolding protein actually degrade close to the N-termini of the polypeptide. As I show you the movie, during the maturation, the N-termini move around. The reason it's move around because you need to shake it off the scaffolding protein. So the scaffolding protein has to get out from the hole of the hexon and then get the hole closed. Now, in order to get after the tail, we have to involve completely different data processing procedure in such a way that we can actually restore the additional protein in one of the 12 vertices in this capsid particle. In this particular case, we have done only around 7 angstrom resolution, and this is a cross-section thing of this density. We actually have 1, 2, 3, 4, 5, 6 different proteins. They're different copies, so there's very complex at this particular portal. I'm just want to next few slides quickly to show you the complexity. So this turn out to be at a 5-fold position, has 12 copies of the portal protein in red and green, surrounded by 10 capsid protein. 10 is not equal to 12. We call symmetry mismatch. So that means each of these proteins are touching the other protein in different domain. Okay, and if we dissect further, we have this thing. I think it makes it simpler. This A1B1 is the capsid protein belong to one hexon. There are 5 of them, so there are 10 capsid protein. And then the blue and the red are the portal proteins. And interesting enough, the domain that interaction of these portal protein to the capsid protein are different. These are the even numbers of the portal protein and the odd numbers of the portal protein. So that means there are multiple domain interactions. Okay, so I think this monomer interaction you cannot derive from a crystallography alone of the protein. In fact, this particular portal protein, the crystal structure was done. But the crystal structure alone could not tell you what types of interaction, what domain, who next to whom, and where. So I think that EM lab will provide you this information. With all these things I described, would just provide you an assembly mechanism. First, turn out to be this particular portal 12 copies of it, form a nucleating site to have some scaffolding protein surrounding it, build additional capsid protein around it, and then more and more, and eventually build the old capsid. And when this is done, the scaffolding has to get out, shaking off from its binding with the N-terminal arm of the capsid protein, and the DNA is encapsulated to build the capsid protein. So I think from this kind of structural analysis help us to build the assembly pathway, how this molecule looks like. So far, all the story I have mentioned to you are biochemically purified virus particle. I mean, that's what we have been doing last 60 years. We call reductionist approach. We're looking at the whole thing, we grind it through, we purify it, and we study it to high resolution. Now, to me, the next excitement in the field, in structural biology and multi-scale imaging, is looking at this kind of virus particle inside the cell. So in the next 5 minutes, I want to show you some of our study looking at the virus particle inside the cell. So here is the microscope. Now, this particular cell may be of some interest to you. This is a photosynthetic bacteria. It's found in oceans, and it captures the sun, convert to organic mass, chemical energy. So its nice thing is very small cell. It's around 0.7 micron. Now, when you look at the electron microscope on the right, it just can't see anything. However, we have employed a particular type of optic called phase plates. It's a Wernicke phase plate, which was, the concept was invented when the microscope was invented many years ago, that if you put a particular device in the focal plane of the objective lens. In such a way, you can shift the phase between unscattered wave and the scattered wave by pi/2. In such a way, resulting the reverse of the phase at the low frequency. So as a result of that, putting a phase space, you immediately see things compared to without the phase space, you don't see anything at all. So it is an immense contrast enhancement. And so this concept was invented, was conceived a long time ago, but had not become practical until more recently because it's a proper material choice. And I think people have the proper nano device and nano fabrication technology. This, doing the phase space is absolutely beneficial in that kind of environment. So, but anyway, so we do this by tomography because no two cell is identical. We can not doing 20,000 cell to average. We would do a 3D reconstruction for each cell. So tomography is by tilting the cell in the electron microscope. And with that, we combine that. And here is we view a 3D tomogram from an individual cell, which has been infected by the bacteriophage. So you can see now I'm sliding up and down with in different Z directions, and there are different features that will jump out to you. Like for example, here there's a virus-looking particle in here, and this is a cell membrane, and there turn out many other things which I'll explain in a minute. This particular one is particularly of interest. This is about 1,000 angstrom round thing, and it's being interpreted as called carboxysome. Carboxysome is a particle that contains the carboxylase, which is an enzyme that fixes the carbon dioxide in the dark reaction of photosynthesis. So carboxysome is the most abundant enzyme on Earth. It's of central interest in energy production, for bioenergy production. It turned out in the photosynthetic bacteria, they all cluster into a particle. The reason they do that is to enhance the efficiency to sequester the carbon dioxide. Dioxide everywhere, but that happens out to be a sequester, and they form a very organized particle. So anyway, we can see all them things in here, and from there, what we do is to annotate this in different features. We can also see that in the bottom rendering means we see each of the individual pixel, and you see there's some virus particle in the outside, and now I'm flipping around, and then eventually I'm going to color code it of different features that I understand. And so here, for example, as in the outer membrane, the cell wall and the inner membrane of the cell. The green one is the follicle membrane, and the blue one is the carboxysome, as I mentioned a while ago. There are plenty of ribosome in here, and then there's one vesicle out here, and then the red one are the infecting phage particle, and then there are other phage particle we call the progeny, the newly synthesized phage particle. They all can be seen in one tomogram. And you can see in here, I'm going to show you one particular phage particle, which is very interesting because like right here, it looks like there is a density penetrating through the cell wall of the bacteria. When you think about that, the virus contains the DNA. The DNA has to deliver into the host. In order to get that delivery safely, they have this tunnel, which is protein, so the DNA can penetrate through it. Now, if there's no tunnel, there's a lot of enzyme in the periplasma which chew up all the DNA before you get into the cell. So we can actually visualize this particular tunnel in the outer cell wall. Now, interesting thing is about there's additional density sticking out. This virus is very unusual. It has an additional, we call horn protein protrusion on the opposite end of the tail, okay? And so from this picture, it's quite clear. This horn has nothing to do with the binding to the cell surface, the phage tail, as all the other bacteriophage. So that also answers the question about the biological role of this horn protein sticking out being for different reasons, but not to do with the cell attachment. So these are the tomograms we're able to get. And from here, then we ask the other, you see here, there are actually some ribosomes leaking out because this is a late stage of the infections. And so now, one of the nice things about doing this tomography, you can do at different physiological states. So we have been able to capture what we call uninfected state. Early stage is only the phage particle outside, and then intermediate stage, and then this final late stage where more phage particle being multiplied in the bacteria. So from here, the next step that we do was to do another data processing of the particle inside the tomogram. In other words, these purple color objects, it's a 3D tomogram. We can actually extract it in 3D volume on each of these we consider the phage particle. And after that, then we have all these subvolume which are particle-like, and we can subject a multivariate statistical analysis and also classification to classify all these particles. And interesting enough, we were able to classify 5 classes of particles in these kinds of tomogram. One is to look like the normal phage with the tail and the horn. The other has only the tail only. The third one, there's no horn and no tail. The fourth one, there seems to be empty inside. And the fifth one seems to be round and smaller. So I think that allows us to consider this may represent fine intermediate assembly of the phage particle in the cell. Okay, now interesting enough, biochemically, we were able to purify only this mature variant. We were not able to purify any of those in quantitative studies. So that signifies the future structural biology. Maybe we turn into the cell. Forget about doing biochemistry. Okay, just looking in the cell, just improve the imaging technology, improve your computing power to get the structures of this intermediate transient or short-lived assembly process. And another interesting thing is we see this particle has only the tail without the horn. That immediately suggests the horn is the last part to be assembled. Okay, you also ask the question of the assembly process. What the sequence of assembly? And then the next point that we discover is we see a density in one of the vertices. Now that is a stage that already expand from this roundish thing, but it's not full with the DNA yet, because there's always a question in the mind of the biology. When does this particle expand? Do all the DNA has to get inside before they expand, or it expand at the early stage? In looking at these structures, we will conclude that expansion actually occurs quite early during the encapsulation process. It doesn't need to be a lot of DNA get into there before expansion. And then secondly, we see this particular density here. We interpret as a terminase, because when the DNA is encapsulated, it needs energy. So these terminases actually provide energy. Again, this is a very transient. When the DNA is packaging, this enzyme will disappear. We've never been able to capture a state like this. And finally, that we allow all these tomographic analysis allow us to get this pathway. So think about that. After the human genome sequences, what is next? The next is discover the biological processes in life. According to estimate, there may be around 200 to 300 biological processes. So I think the excitement in structural biology, to me, in the next 10, 20 years, is to discover structure of these processes inside the cell. I think tomographic analysis, which is still in a very early stage, will provide that opportunity. And finally, I don't want to bore you, we work on not only phages, but herpes, and we look at herpes, infect the cell, we do the tomography on that, and then we actually discover that actually the herpes, we can purify it. What we see structurally in the highly purified one, actually some portion of it don't look the same as in the cell. So that actually, again, in order to do a validation check, we need to do the structure in the cell, like in this case on the herpes. And finally, I'd like to summarize what I told you today, that the cryo-EM can determine atomic resolution of protein component virus at different physiological state, before DNA come in, after DNA come in. Secondly, I also show you some of our latest work in tomography, which give you a different structural snapshot of the assembly process of infections inside the whole cell. And finally, I'd like to acknowledge my collaborators and colleague Rick developed the entire suite with Paul Adams in Berkeley on the model building, and Donghua did the imaging, and Wei Dai did the tomography. And this in long-term collaboration with John King at MIT and Powell Christopher, who worked with us on developing the model building in real space, and then Rickson on the herpes project, and Nakayama from Japan, who introduced us this faceplate technology. Thank you very much.
So, questions? Chris.
Yeah, I have a question. One of the things, thanks. So beautiful presentation, first of all, and really neat to see both kind of pushing on the structure determination and on the in-cell imaging. I have a question concerning the different conformational states. You didn't really talk about that very much in your presentation, where you have different molecular conformations and how that scales with size of subcellular assemblies. Do you have any feeling for that?
Yeah, so I think I showed it between the procapsid and the mature variant. They actually conformational differences. And so one thing I skip very quickly because of the interest of time in the case of the herpes. And I think we actually discover that in some of the herpes, this particular part in the penton part is actually losing one of the penton. Okay, so that means after the herpes get inside the cell, they may have undergo different types of conformational variation. So in other words, how many herpes it takes to make you sick? Okay, we don't know. I think this is an interesting question because the virology is now studying virus. The new trend of virology is actually virus and host interactions. And then in the DOE setting, you got a community, different hosts, how one host talk to the next host. So I think this providing information at whatever resolution level about the structural variation. So I think the common theme in the future in tomography is the structural diversity of the cell molecules. And I think the question is, why are they different? And that may affect its well-being and how it affects all the other processes in the cell, the homeostasis and how we get sick and not get sick. So that, to me, that what we may possibly bring to the tables.
Yeah, thank you for the beautiful presentation. Even I understood it. So. Somewhere in your talk, you mentioned capsid expansion and contraction. And what I was wondering was whether that was a real intermediate steps that you have solved at every step, or was it a model that you showed?
No, those are experimental data. It's not a model.
Right.
And then I substantiate with the tomogram, we actually see two different sizes.
What is your time resolution?
No, those are not time resolution. In other words, we can biochemically purify a mature variant, and we do structure. We can actually run a density gradient gel because they have no DNA, so you run a different band. We can purify that one and do the second structure. So it's not a time. So what we don't do is time. But by looking at the structure, we can deduce the time elements of it.
Thank you.
There you go, sir.
Great talk. So one of the big advantages of the cryo-EM is solution studies, really, and you can capture multiple states. Right? So in your case, P22, you have the prophage and you have these different kind of thing. Now, are there any intermediates between them?
Yes, absolutely. So this is what I saw on the tomogram study. In here, there are 5 states. Okay, so that is why I'm excited about tomography, because I can't purify it. Okay, but they're in the cell, we just look.
So another question, why? Because the virus is so symmetrical, but there's only one entry of this DNA transfer apparatus.
Yeah.
Why there is only one entry?
Okay, so there is only, so because it takes energy, it's no free meal. Even the DNA comes in, it's their energy. So their particular enzyme, as we pointed out here, this little thing, the terminates actually provide the energy. So in other words, you can think about it. If you have a water hose, water your garden, right? If you have multiple water hoses, it's very confusing. But if you put your water hose orderly, you can easy in and easy out, because they have to think about how to get in and how to get out effectively. So I think now the scaffolding protein is different. The scaffolding protein, they get out, we believe they get out from all the hexon, because there's a close exit, you get out here, get out there. But there's only one way for the DNA.
So the tomographic approach is really pretty cool to see the different states. But I want to go back to the previous one with the phage where you showed the maturation. How does that compare to the work that Jack Johnson did on HK97 with X-rays? Are they relatable?
So I want to point out the difference between our case and the Jack Johnson case. Jack Johnson is actually recombinant particle. It's not infectious particle. Okay, so
they did it on our beamline. Yeah, it's not infectious.
yeah, not infectious. That's why I get into here, right? So our is an infectious particle. Okay, so that's one important difference. That is why David Stewart in Oxford spent millions of British pounds to be able to diamond light sources to do this infectious stuff. Okay, and that was very interesting because the fold, what we see is similar to Jack Johnson's fold in the Hong Kong 97.
Michael?
Hi. A little bit different, but some photosynthetic bacteria, cyanobacteria, contain gas-containing vacuoles that keep them buoyant in the water, and those can be collapsed and they sink. Some of those may contain hydrogen gas. One could imagine that such a material might be leaky, but such a material might be of interest for lightweight hydrogen storage. I'm curious why one rarely sees anything in the literature about that. Are you aware of people investigating that kind of system, perhaps with EM or so?
No, not yet. I'm going to. Because the reason I stumble in that, because some DOE guy talked to me 10 years ago and say, why don't you work on something useful for DOE? And I said, okay, what's useful? That's why I stumble in, I look up the web and find out there's a Professor Penny Chisholm at MIT. That's why I get into these cyanobacteria. But then when I work on it, it turned out to be a paradigm because this cell is small, smaller than the E. coli. E. coli is big compared to these kinds of cells. So I'm actually just studying the carboxysome is interesting because the Christopher, Yates from UCLA. I mean, one thing I didn't talk about, if you look inside the cell, the carboxysome has different sizes. Okay, and one of the interesting questions is how the carboxylates are organized inside the carboxysome. I think we have the answer, but we'll tell you next time. And so, because of different sizes, and there's no hope to do crystallography on that, I think. Okay, unless you really purify it and so forth. And so I think the interesting question is the biogenesis of this kind of carboxysome, how they put together and to make it fixing the carbon dioxide. So I think some of the kinds of bacteria you mentioned to me today is really interesting. I really want to look at it. One of the things, just looking at the thyroid membrane, you see, this is the green bit. I mean, there's tremendous excitement, I think. The photosynthetic reaction center and so are done. But if we put that in that context, we'll be filling the gaps in the future. And so I think these are, looking at some of the vacuum and phagobutysome that you have done before, I think would be right on the target for the future specimens. Okay, good. Thanks so much.